Right now, in a laboratory somewhere in the world, a surgeon is performing a highly complex biopsy. And the stakes of this procedure are, well, they're absolute.
Yeah, they really are.
But the thing is, their scalpel is made entirely of invisible light. And their surgical theater is just a microscopic drop of fluid.
Right.
Their patient is an organism that is actually smaller than a single grain of salt.
Hmm.
We are entering this surgical landscape that is completely invisible to the naked eye. We're working at the absolute limits of human perception here.
It forces us to completely reevaluate our definition of physical manipulation.
Oh, absolutely.
We are operating in this realm where fluid dynamics, the viscosity of water, and cellular tension are what dictate every single movement. The physics at this scale are just entirely alien to our everyday experience.
Well, welcome to the premiere of Season 2. This is episode 16 of our journey inside the IVF lab, and as we begin this deep dive, I really want us to deliberately slow down.
Yeah, that's a good idea. Take a breath.
Exactly. We need to ground ourselves in the monumental scale of the journey we've taken so far. I mean, just think about the timeline for you listening. We started the series by looking at a single unfertilized cell resting in a dish.
Just one cell.
Just one. We observed the precise, quiet biology of maturation. We watched that cell evolve from an immature oocyte, navigating delicately through the M1 phase, and finally achieving that vital M2 stage of maturity, which is the biological prerequisite for creating life.
Right. Getting to M2 is a massive biological hurdle.
It is. And then we followed that one cell as it was fertilized, as the pronuclei fused, as the solitary cell cleaved into 2, then a 4, then 8. And now we're looking at an incredibly complex day 5 structure, a fully expanded blastocyst sitting right there in the laboratory incubator.
It represents a profound architectural transformation. I mean, from a quiet, solitary genetic vessel to this dynamic, rapidly dividing organism that is actively organizing its own cellular future.
So our mission for this deep dive today is to give you a comprehensive, scientifically rigorous mental map of the pre-implantation genetic testing biopsy, or, what's commonly known as PGT.
PGT, right.
We are going to map out the reality of embryonic microsurgery because it's an environment governed by strict genetic protocols, thermal limits, and intense spatial awareness.
And for you listening, whether you're an embryologist in training trying to, perfect the spatial memory of your hand movements, or a clinical nurse tasked with explaining this highly technical timeline to families.
Which is not an easy job.
Not at all. Or maybe you're a patient anxiously waiting for your eutloid genetic results. This deep dive is meant to pull back the curtain on the laboratory for you. We are stripping away the mystery.
Replacing the anxiety of the unknown with a deep understanding of the managed structural precision that happens behind those closed laboratory doors.
Exactly.
So before we even think about powering up an infrared laser or bringing glass micropipettes anywhere near this organism, we have to map the territory.
We need the blueprint.
Right. We need to understand the precise anatomy of a day 5, 6, or 7 embryo, because performing a biopsy blindly is impossible. We have to know the exact biological geography to know where it is actually safe to extract cells.
And the underlying directive of a PGT biopsy is clinical efficiency and risk mitigation. The laboratory is tasked with identifying which embryos possess the correct, balanced complement of 46 chromosomes.
What we term a euploid state.
Right, euploid.
Yeah.
By transferring a euploid embryo, the clinical objective is to drastically reduce the statistical likelihood of miscarriage and, shorten the patient's timeline to a successful healthy pregnancy.
So to visualize what we are actually looking at under the microscope, I want you to imagine the blastocyst as a highly pressurized soccer ball.
Oh, I love the soccer ball analogy.
It works so well, right? So the air inflating the inside of that ball is analogous to a fluid-filled cavity within the embryo. That's called the blastocyst.
The blastocyst, yeah.
And the leather panels making up the outer surface of the ball, those represent a distinct layer of outer cells. But here's the key part. Tucked away securely, attached to the inside wall of one of those outer panels, is a tiny, dense prize package.
And the architecture of that soccer ball is the result of the very first major differentiation event in human development.
Meaning the cells are finally picking a job.
Exactly. Prior to this stage, the embryo cells were totipid. Essentially, they were capable of becoming absolutely anything. But by day 5, the cells have segregated into 2 strictly distinct biological lineages.
Okay, so let's break those down.
Sure. So that dense prize package inside the soccer ball, that is the inner cell mass, or the ICM, Those are the pluripotent cells destined to form the actual fetus.
Leaving the leather panels on the outside to become the support structure.
Right. Those outer cells form what we call the trophectoderm layer. They're destined to become the placenta, the amniotic sac, the protective membranes. They will invade the uterine lining and establish the maternal-fetal connection. But, and this is crucial, they will never form the baby itself.
Never.
Never. Recognizing this strict, impenetrable biological boundary is the entire foundational premise that makes an embryo biopsy scientifically viable and medically safe.
And this brings us to a pervasive and frankly very intuitive fear that many patients harbor when they first hear the word biopsy.
Oh, absolutely.
I mean, the thought process is completely logical. If a clinician is surgically removing cells from an embryo, aren't they risking structural damage?
Right, like are you taking a piece of the baby's arm?
Exactly. Could extracting those cells cause a missing limb, a physical deformity, or some neurodevelopmental defect in the child?
It is a totally rational fear if you visualize the embryo as a miniature, fully formed human. But at the blastocyst stage, there is no physical body. There's only a map of potential.
Just a map.
Right, and the clinical reassurance here is absolute. The embryologist never touches, approaches, or alters the fetal-forming inner cell mass. The ICM is considered a strictly restricted zone.
Do not touch.
Do not touch. We are exclusively interacting with the trophectoderm, the future placenta.
But wait, how does testing the placenta tell us about the fetus? If they are differentiating into two different structures, why do we trust the outer cells to speak for the inner cells?
Well, we rely on their shared biological origin. Both the trophectoderm and the inner cell mass descended from the exact same original fertilized egg.
Oh, right, so they have the same starting blueprints.
Exactly. They share the same initial genome. So we extract a minute sample, typically just 5 to 10 cells from that outer trophectoderm layer. And because of their shared genetic lineage, those trophectoderm cells serve as a highly accurate proxy for the chromosomal makeup of the inner cell mass.
It's effectively like reading the blueprints of a house by examining a brick from the garden wall.
That's a great way to put it.
Okay, so with the biological target established, we really have to deal with the staggering physical reality of the procedure. We're trying to mechanically separate a 5-cell cluster from an object that is approximately, 100 to 120 microns in total diameter?
Yeah, right around there.
For context, a single strand of human hair is about 70 microns thick. So how on earth do you build a surgical theater for something that small?
You construct what is essentially a highly controlled microarena. The embryologist prepares a specialized, optically clear biopsy dish.
Okay.
But within this dish, they don't just flood it with fluid. They pipette discrete, microscopic droplets of buffered culture media.
And this media isn't just salt water.
No. It's a highly sophisticated, nutrient-dense solution. It contains specific amino acids, energy substrates like pyruvate and lactate, and critical pH buffers. It is specifically designed to mimic the biochemical environment of the human fallopian tube and uterus.
Wait, if you place a microdroplet of fluid under the intense, focused heat of microscope stage warmers and optical lights, wouldn't that droplet simply vaporize into the ambient air within seconds?
Yes, it would.
And you'd have a massive spike in osmolality as the water evaporates and leaves all those salts behind.
It would be catastrophic. The salt concentration would spike, the pH would swing wildly, and the embryo would undergo rapid osmotic shock and die.
So how do you stop that?
To prevent this, every single microdroplet is submerged under a thick layer of ultra-pure mineral oil.
Oil and water don't mix, so the oil just acts as a liquid lid.
Exactly. It is an impermeable thermodynamic shield. The oil layer completely prevents aqueous evaporation. It also acts as a thermal buffer to maintain a perfectly stable 37 degrees Celsius.
Wow.
And it regulates gas exchange, specifically keeping the carbon dioxide and oxygen concentrations within the microdroplet locked at optimal physiological levels. So the oil maintains the stasis of the arena.
Okay, so the arena is secure. Let's look at the hardware required to actually peer into it, because this isn't your standard high school biology microscope where the lens points down at a glass slide from above.
No, definitely not. We're using an advanced inverted microscope.
Right.
With an inverted microscope, the high magnification objective lenses are actually housed underneath the mechanical stage, pointing upward to the bottom of the glass dish, and the light source and the condenser are mounted above, shining down,
leaving the entire top and sides of the dish completely unobstructed, which is essential because we need physical access to bring our surgical tools down into the droplet.
Exactly, and those tools are manipulated using a dual micromanipulator system.
Okay, let's talk about the mechanics of those manipulators. Because you are dealing with a target of 100 microns. The human hand naturally possesses a physiological tremor.
Everyone's hand shakes a little.
Right. Even the steady hand of a neurosurgeon has a slight, imperceptible vibration. A single heartbeat transferring down your arm would send a glass micropipette crashing completely through the embryo and shattering on the other side.
It would just obliterate it. You cannot simply hold these tools with your fingers.
Right, so what I'll do?
Well, the physical limitations of human physiology necessitate mechanical intervention. The micromanipulators are sophisticated gear reduction systems. They often utilize hydraulic, piezoelectric, or highly geared motorized components.
Okay, so they scale down your movements.
Exactly. They act as a mechanical translation interface. The embryologist rests their hands on these large ergonomic joysticks. And when the embryologist moves the joystick a full centimeter, the macro world, the micromanipulator translates that into a smooth, frictionless movement of a single micron inside the microdroplet.
Wow, so it just filters out the human tremor entirely and scales down the intent.
Perfectly said.
So on the left side of this mechanical setup, we have the holding pipette. It's a glass capillary with a blunt fire-polished tip. Its sole function is to act as an anchor.
Right.
And on the right side, we have the biopsy pipette. This one is entirely different. It's open-ended with sharp, precisely beveled edges designed specifically to cut and collect the cellular sample.
And integrated directly into the optical path of this microscope, firing straight through those upward-facing lenses, is the primary surgical instrument,
Which is
an infrared medical laser.
a laser, firing microscopic pulses of heat into a droplet of water containing an embryo. I mean, the margin for error seems virtually non-existent. How do you even guarantee the laser is striking exactly where the crosshairs on the screen indicate? If the alignment is off by a fraction of a millimeter, couldn't you just obliterate the inner cell mass?
You absolutely could.
Yeah.
Which is why the calibration protocols are obsessive. I mean, before a patient's embryo is ever removed from the safety of the incubator, the embryologist must perform a rigorous laser alignment using a specialized reference slide.
So you're practicing first.
We aren't just looking at the screen. We are actively firing test pulses into a synthetic medium to measure the exact geometric diameter of the physical hole it creates.
Wow, checking the literal burn mark.
Right. And we verify the optical focal plane to ensure the energy isn't converging too high or too low in the fluid column. We calibrate the pulse duration down to the absolute microsecond.
So the spatial coordination and the software fail-safes are layered.
Heavily layered. If the beam deviates by even a fraction of a micron, the clinical software flags the discrepancy and physically locks the system.
Okay, so the environment is stabilized, the manipulators are scaled, the laser is perfectly calibrated, the surgical theater is ready.
It's go time.
We bring the dish containing the blastocyst out of the incubator and place it onto the heated microscope stage. This is where the physical approach begins.
And this is where the tension spikes. The embryologist locates the blastocyst through the eyepieces. And from this moment forward, every single movement is deliberate, utilizing those hydraulic joysticks.
Okay.
The left holding pipette is brought into the microdroplet and positioned adjacent to the embryo. Then, the embryologist applies a very slight, highly regulated negative pressure, a microvacuum, through the pipette.
But it doesn't suck the whole embryo inside, right? Because the pipette opening is too small for that.
Exactly. It just grabs the outer shell, acting like a tether to lock the sphere in a fixed position against any fluid currents.
Got it. Once tethered, we have to establish the geography of safety. The orientation of the blastocyst at this precise moment dictates the safety of the entire procedure.
Right, because we need to find the prize package.
So using the glass tools, the embryologist gently, physically rolls the blastocyst around on the tip of the holding pipette. They are visually hunting for the inner cell mass.
We have to position the fetal cells as far away from the laser's focal point as the geometry of a sphere will allow.
So what's the standard protocol for that?
The protocol is to rotate the blastocyst until the dense cluster of the inner cell mass is resting securely at the 7 or 8 o'clock position on the visual field.
Tucked safely behind the bulk of the embryo.
Yes, and pressed lightly against the blunt holding pipette.
Leaving our target zone a healthy, expansive stretch of trophectoderm cells completely exposed on the opposite side, which is typically floating freely around the 1, 2, or 3 o'clock position.
Exactly. So with the geography locked, the embryologist initiates what we call the draw.
The draw.
The right pipette, that sharp, open-ended biopsy tool, is advanced toward that 3 o'clock trophectoderm. The embryologist applies a fine, highly controlled pneumatic suction to the right joystick, and they gently draw a microscopic loop of those outer cells into the internal channel, the lumen, of the biopsy pipette.
Okay, so you have a microscopic section of the soccer ball's leather panels pulled slightly inside the glass tube.
Yes.
The cells are under mechanical tension now, stretched tight across the sharp opening of the pipette. And this is the moment we introduce the infrared laser. Let's really delve into the actual physics of this laser, because we are talking about a non-contact diode laser operating at exactly 1480 nanometers.
That specific number is so important.
Why that specific wavelength? I mean, why not ultraviolet or a standard surgical CO2 laser?
The choice of 1480 nanometers is honestly a masterpiece of biophysics. At that specific infrared wavelength, the photon energy perfectly matches the vibrational frequency of the oxygen-hydrogen bonds in water molecules.
Oh, which means the laser energy is targeting the water, not the cellular tissue itself.
Exactly. When the laser fires, the infrared energy is absorbed almost instantaneously by the water molecules trapped in the microscopic spaces between the cells.
The cell-to-cell junctions.
Right. And the water in that highly localized focal point rapidly heats and vaporizes. It creates a microscopic steam bubble.
So it's boiling it.
Briefly, yes.
Mm-hmm.
And the rapid expansion and collapse of that steam bubble creates an acoustic mechanical force that just snaps the protein bonds holding the cells together.
Wow. So it's essentially using localized phase change water turning to steam to physically blast the tight junctions apart rather than burning the cell membranes with thermal radiation?
That is the genius of it.
Mm-hmm.
And because the energy is so perfectly absorbed by water, the thermal penetration depth is incredibly shallow. The heat dissipates into the surrounding culture media almost immediately.
So there's no widespread burn radius.
Virtually zero thermal collateral damage. The adjacent cells just a few microns away remain completely unaffected.
Okay, so the embryologist steps on the foot pedal, the laser pulses, but it's not a continuous beam, right? Like a lightsaber.
No. It fires in ultra-short microsecond bursts, faster than the blink of an eye. We use the absolute minimum number of pulses required, usually just 2 to 4 rapid flashes, aimed precisely at the stretched cellular junctions right at the edge of the glass pipette.
So it's acting less like a knife slicing through butter and more like a tool creating a weakened, perforated edge along the cellular tension line.
Yes, a perforated edge is exactly what it is.
But the laser alone doesn't usually finish the job, right? There is a mechanical component to the extraction.
Correct. The laser compromises the structural integrity of the trophectoderm layer. But once that perforated edge is established, the embryologist executes a delicate mechanical maneuver.
What kind of maneuver?
Well, they might create microscopic friction by gently rubbing the sharp edge of the biopsy pipette against the blunt edge of the holding pipette. Or they might utilize a swift microscopic flicking motion with the right joystick.
And that just pops them off.
Right. This mechanical force pairs the sample cleanly away along that weakened perforated line.
And the 5-cell cluster snaps free.
It does.
The biological separation is complete. We now have a microscopic piece of the future placenta isolated inside the biopsy pipette. But extracting the sample from the embryo is truly only half of the laboratory battle.
Honestly, it might be the easier half.
Really? Because now we are faced with an entirely new and arguably more stressful challenge: securing that DNA without contaminating it.
This is where the atmospheric tension in the IVF lab shifts entirely. I mean, the laser dynamics are governed by predictable physics, but the highest risk of critical failure in a PGT biopsy does not come from the laser. The most pervasive threat is ambient environmental DNA.
Okay, let's visualize this transition. The laser sequence ends, the holding vacuum on the left pipette is released, and the parent blastocyst is gently moved to a safe resting drop on the far side of the dish.
Right, the parent is safe.
But what is happening with the embryologist's visual focus during all this?
Oh, the embryologist maintains an unbroken continuous visual lock on the tiny extracted sample.
They don't look away at all.
Never. That 5-cell cluster is nearly translucent. If you look away from the eyepieces, if you lose track of its position within the vast three-dimensional space of that microdroplet, finding it again is agonizingly difficult.
So keeping the sample in sight, the operation moves away from the microscope stage. We transition from the arena of microsurgery into what is fundamentally a specialized cleanroom environment. The sample is transferred to the tubing station.
And the tubing protocol demands a dramatic escalation in environmental control. The station is housed within a dedicated laminar flow hood.
What does that do?
It's a workstation that utilizes high-efficiency particulate air, or HEPA, filters to bathe the workspace in a continuous flow of ultra-purified air, actively pushing any ambient particles away from the sample.
And the people.
The embryologist must don full protective gear, fresh surgical masks covering their mouth and nose, hairnets, and freshly donned sterile, powder-free gloves.
To someone outside the field, deploying hazmat-level protective gear over a cluster of 5 cells might seem a little paranoid. Why is the lab so terrified of contamination?
Because of the staggering analytical power of the genetic sequencing laboratory that will receive this sample.
The external lab.
Right. The sequencing equipment utilizes a biochemical technology called polymerase chain reaction, or PCR.
PCR, right.
PCR is essentially a molecular photocopier. It takes the vanishingly small amount of DNA present in those 5 cells and exponentially amplifies it. It multiplies it millions and millions of times until there is a large enough physical volume of genetic material for the sequencing lasers to actually read.
But PCR is completely blind, isn't it? It just amplifies whatever genetic material happens to be inside the tube.
It is entirely indiscriminate. If a single squamous epithelial cell, a microscopic flake of skin shed from the embryologist's wrist, drifts into that tube,
Oh, wow.
or if a microscopic droplet of aerosolized saliva from a breath bypasses the mask, or even if a dust mite carrying its own DNA lands in the sample, the PCR process will aggressively amplify that contaminant DNA alongside, or even preferentially over, the embryo's DNA.
Which means when the genetics lab runs the test, the resulting report might map the chromosomal profile of the embryologist's shed skin cell rather than the genetic health of the embryo.
Exactly. The sample would be overwhelmingly contaminated. The result would be entirely uninterpretable, rendering the entire surgical biopsy useless.
So all that laser work for nothing.
For nothing. And the clinician would have to inform the patient that no data was obtained. The only recourse would be to potentially thaw the embryo in the future, perform a second biopsy, and refreeze it, a process that introduces significant cumulative stress and risk to the blastocyst.
So the uncompromising paranoia of the cleanroom dynamics is literally the only way to prevent that cascade of failure.
It's the only way.
So to guarantee that absolute purity, the embryologist doesn't just take the cells straight from the biopsy dish to the testing tube. The sample has to be aggressively washed.
Yes. The washing sequence relies on the mathematics of serial dilution.
How does that work?
The embryologist uses an ultrafine, sterile glass capillary tube, controlled by mouth pipetting, or a highly sensitive microsyringe, to pick up the 5-cell cluster.
Okay.
They move the cluster out of the biopsy fluid and deposit it into a pristine droplet of sterile wash buffer. They then pick it up again and move it to a second, fresh droplet, then a third, and often a fourth.
So by physically transferring the cells through multiple sterile pools, they're systematically diluting and stripping away any background proteins, any residual culture media, and crucially, any fragmented cellular debris generated by the laser strikes.
Exactly. We have to leave all of that behind. And the final step is the microscopic load.
Getting it in the tube.
Right. The embryologist draws the cleanly washed cells up into the very tip of the capillary tube. But they must draw it up in a fractional volume of fluid.
Why fractional?
The genetics lab requires the cells to be delivered in virtually zero liquid because excess buffer can actually inhibit the biochemical reactions of the PCR process. We're talking about loading the cells in less than 1 microliter of fluid.
Okay, let's contextualize that volume for a second. A standard single drop of rain falling from the sky is approximately 50 microliters.
Right.
We are dealing with less than 1/50th of a single raindrop.
It's almost nothing. The embryologist carefully lowers the capillary deep into a tiny sterile PCR tube and expels that microscopic fraction of fluid directly onto the absolute bottom of the tube.
But at a volume of less than a microliter, human tactile sensation isn't enough to know if the droplet actually left the glass capillary. You can't feel it. You can't just assume it went into the tube.
No, assumption is not a laboratory protocol. Ever.
So what do?
The embryologist places the loaded PCR tube under a specialized high-power stereo microscope. They adjust the lighting to illuminate the curved bottom of the plastic tube, looking straight down the barrel.
Okay.
They must visually confirm, actually see the light refracting off the 5-cell cluster, floating in that microscopic droplet at the very base of the tube before they can confidently seal the cap.
Wow. Okay, so the DNA is now sealed in its tube. The parent embryo remains resting in a dish across the room. We have successfully extracted the genetic material, but this introduces a massive overarching logistical vulnerability.
The waiting game.
The genetic sequencing takes time. The tubes will be shipped to an external reference laboratory, and the sequencing results won't return to the clinic for several days, sometimes up to 2 weeks.
2 weeks is a long time in a lab.
During that massive time gap, the embryo and its specific genetic data are physically separated.
And this is the chain of custody challenge. It is the administrative nightmare that keeps lab directors awake at night.
Ugh.
I mean, if you mix up the tubes, if you assign a euploid normal result to the wrong embryo, the entire foundation of IVF collapses.
Everything is built on trust that the sample matches the embryo.
Everything. The lab must guarantee with unbreakable certainty that the DNA sample and the parent embryo remain perfectly matched across time and space.
So technology obviously provides the first layer of defense here. The laboratory relies on microscopic barcodes printed on the side of the PCR tubes. But dealing with labels on tubes that are barely larger than a pencil tip, which must withstand being packed in dry ice, I mean, that is a profound engineering feat in itself.
The cryo-safe labels are vital, sure, but when the clinical stakes are this absolute, technology alone is insufficient. Software can glitch, printers can misalign.
Right.
So the laboratory relies on an uncompromising, human-enforced firewall. It's known as dual witness verification.
Walk us through what that actually looks like at the tubing bench.
Okay, so before the embryologist ever dispenses that cellular sample into the PCR tube, the entire procedure halts.
Everything stops.
Everything. A secondary embryologist must physically stop their own work, walk across the laboratory, and stand directly beside the tubing station.
They act as the witness.
Right. The first embryologist reads aloud the patient's unique identification number, the embryo's numerical designation, and the specific barcode on the dish. And the secondary witnessing embryologist visually inspects the microscopic barcode printed on the DNA tube and verbally confirms that every single alphanumeric character matches perfectly.
It is a mandatory, highly structured pause in the workflow, a human check against administrative drift.
Exactly. And once the verbal and visual verification is complete, the data matrix, the exact tube number, the cell count, the embryo number, and the patient identifiers is logged simultaneously into the laboratory's secure electronic database and physically written onto the transport manifest that will travel with the samples.
And only then is the sample loaded.
Only then.
Okay. Meanwhile, what is the biological status of the parent blastocyst? Because we have fired a laser at it, we have mechanically torn away a piece of its outer trophectoderm shell. It seems highly unlikely that it just sits there unaffected.
No, it doesn't. There is a dramatic physiological response that patients are rarely aware of. Following the biopsy, the blastocyst will undergo a temporary collapse. Yes. The laser breached the tight cellular junctions that maintain the pressure of the internal fluid cavity. So without that seal, the fluid leaks out and the embryo deflates. It loses that expanded spherical shape we discussed earlier.
It sounds alarming, to be honest, like a balloon losing its air.
It looks alarming,
Yeah.
but it is a completely normal, anticipated biological response. The cells themselves remain highly viable.
Oh, really?
Yeah, in fact, many embryologists intentionally collapse the blastocyst prior to freezing, as removing that water content actually prevents lethal ice crystal formation during the freezing process.
Oh, that makes perfect sense. Which leads directly to our next step. Because we obviously cannot leave a deflated blastocyst sitting in a warm incubator for 2 weeks, waiting for the genetic report to return.
No, its metabolism would continue, it would attempt to hatch, and it would ultimately degrade.
We have to hit the pause button on its biology.
We do. The biopsied embryo is immediately transferred to the cryopreservation bench.
Okay.
It undergoes the precise, highly toxic vitrification sequence we covered extensively in a previous deep dive. It is exposed to concentrated cryoprotectants and plunged into liquid nitrogen.
Instantly dropping its temperature to minus 196 degrees Celsius.
Right, and at minus 196 degrees, all cellular metabolism ceases. Time effectively stops.
So the physical divergence of our two entities is now complete. The parent embryo rests safely, suspended in a glassy state within a liquid nitrogen cryotank. Meanwhile, its corresponding DNA tube is packed tightly on dry ice and handed to a medical courier, shipped overnight to a specialized genetics laboratory.
The intense physical manipulation within the IVF lab is finished, but the intellectual diagnostic journey is honestly just beginning.
Right, because weeks pass. The genetics lab runs the PCR amplification, they sequence the DNA, and they send a highly detailed data report back to the clinic's electronic medical record.
And this is where it gets complicated.
Right, because when the physician and the patient sit down to read that report, the results are rarely a simple binary pass or fail. The data presents profound nuances. What does this genetic readout actually represent?
Well, to interpret the data accurately, we must confront the diagnostic horizon. We have to ruthlessly remind ourselves of the mathematical and biological limits of the procedure we just performed.
The mathematical limit being the sample size. We took a microscopic snapshot. We surgically excise roughly 5 trophectoderm cells out of an embryo that likely contains over 100 or 150 total cells at this stage.
Exactly. The entire clinical premise relies on the assumption that those 5 sampled cells accurately, flawlessly represent the chromosomal status of the other 145 cells.
Specifically, the crucial cells of the inner cell mass.
Right, and most of the time that assumption holds true. Biology is remarkably consistent, but embryonic biology is also incredibly complex, and this brings us to a massively important diagnostic phenomenon known as mosaicism.
Okay, let's define mosaicism. Because if you think of a mosaic piece of art like a floor or a wall made up of hundreds of tiny different colored tiles, I think it gives us a great visual.
It is the perfect biological analogy. Mosaicism is a diagnostic reality where a single embryo contains a mixture of 2 or more genetically distinct cell lines.
2 different sets of genetics in one embryo.
Yes, in the context of PGT, it means the embryo is not uniformly uploid or uniformly aneuploid. It possesses a mosaic of both.
Okay.
It has some cells that are completely chromosomally normal, possessing 46 chromosomes, and it has some cells that are abnormal, perhaps carrying an extra chromosome or missing one entirely.
But wait, how does an embryo even end up with 2 different sets of genetics? Didn't all start from the same egg and sperm?
It did. The original fertilization event might have been perfectly normal, but as that single cell began to divide 2 cells, 4 cells, 8 cells, a post-zygotic mitotic error occurred.
A mistaken division.
Exactly. During one of those early cell divisions, the chromosomes didn't pull apart evenly. One daughter cell receives an extra chromosome, the other was left short. Okay, I follow. From that moment on, every cell that descends from that specific erroneous cell will carry the abnormality, while the cells descending from the normal lineages will remain healthy. The timing of when that error occurs dictates how widely the abnormal cells are distributed throughout the blastocyst.
Wow. This introduces a terrifying diagnostic variable. I
Hmm.
mean, if you happen to pull your biopsy sample from a patch of the trophectoderm that is heavily populated by those abnormal cells, the sequencing report will flag the entire embryo as abnormal.
Yes, it will.
But what if the rest of the embryo, and crucially, what if the fetal-forming inner cell mass is actually entirely normal?
This is the bleeding edge of reproductive medicine right here. And it forces a profound shift in how clinicians counsel patients.
How so?
Well, years ago, an abnormal biopsy result meant the embryo was immediately discarded. But as our sequencing technology became vastly more sensitive, we began detecting these subtle mosaic profiles. And we are discovering something miraculous about embryonic biology.
That embryos possess an inherent biological capacity to self-correct.
Yes, the clinical data suggests precisely that. We're observing that some mosaic embryos, when transferred into a uterus, possess mechanisms to effectively manage their abnormal cell populations.
How do they do that?
The embryo might recognize the aneuploid cells and undergo targeted apoptosis, essentially instructing the abnormal cells to die off.
Wow.
Or, even more fascinating, the embryo might physically marginalize the abnormal cells, selectively pushing them out into the trophectoderm lineage to become the placenta, while strictly allocating the normal healthy cells to the inner cell mass to form the fetus.
So a mosaic embryo, one that flags as partially abnormal on a PGT report, can actually still result in a perfectly healthy, genetically normal baby.
It happens frequently, and this changes everything about how a patient must read their biopsy results. It underscores the profound clinical reality that a PGT biopsy is an incredibly powerful tool of probability and priority.
It provides the physician with actionable data to prioritize which embryo to transfer first, maximizing the statistical likelihood of success.
Exactly. But it is not a crystal ball.
It is a highly advanced screening tool, not a diagnostic certainty. A normal euploid result is not an absolute 100% guarantee of a live birth, as there are obviously countless other factors involved in implantation.
Right. And conversely, an abnormal or mosaic result is not always an absolute guarantee of clinical failure.
Well, when you synthesize the sheer scale of everything we've explored today, I mean, the physics of the infrared laser, the strict fluid mechanics of the microdroplet, the hazmat protocols at the tubing station, and the incredibly complex biology of mosaicism, I think it all distills down to a central defining concept.
But
In the biopsy suite, the embryologist is really just carefully opening the book of the embryo's genetic story and reading the preface.
just the preface.
We are surgically extracting just enough preliminary data to help navigate the uncertain road ahead while doing everything in our power to ensure the book itself remains completely intact and unharmed.
That is the ultimate goal of the laboratory. We are reading the preface to ensure we are setting the patient on the most statistically promising path, fully aware that the complete narrative of that embryo's potential is yet to unfold.
So let's briefly retrace the incredible microsurgical sequence we've mapped out for you today. We started by orienting the blastocyst, learning how the embryologist establishes that strict physical boundary between the protected, fetal-forming inner cell mass and the target tissue of the trophectoderm.
Right, the restricted zone.
Then we explored the brilliant biophysics of the 1480-nanometer infrared laser, utilizing localized water vaporization to separate cell junctions without causing thermal damage to the surrounding biology.
The microsteam bubble.
Exactly. And we detailed the extreme cleanroom dynamics required during the DNA tubing phase, highlighting how a single stray skin cell amplified by PCR could completely derail a patient's cycle. And finally, we mapped the unbreakable dual-witness barcode protocols that ensure the embryo sitting in the liquid nitrogen tank is perfectly matched to the genetic data arriving weeks later.
It is a breathtaking convergence of biology, optics, and administrative discipline.
It really is.
But looking at the trajectory of this science, I really want to leave you with a provocative thought about the future.
Okay, let's hear it.
Considering the staggering speed at which genetic sequencing technology is advancing, it's becoming more sensitive and requiring less starting material every year. How might future breakthroughs allow us to read this genetic preface without ever firing a laser or removing a single cell?
You are talking about non-invasive PGT.
Precisely. As an embryo grows and actively expands inside its tiny microdroplet of culture media over those 5 days, it naturally sheds microscopic fragments of its own DNA, cell-free DNA, into the fluid surrounding it.
Oh, wow.
The great diagnostic frontier for the next decade of clinical embryology is this. Could the mere fluid an embryo floats in one day hold its entire, highly accurate genetic map? Could we simply aspirate the spent culture media, sequence the shed DNA, and render the entire concept of the surgical embryo biopsy completely obsolete?
Reading the water instead of taking the cells, it would fundamentally change the risk profile of IVF forever.
It would.
That is a truly fascinating horizon to watch. Thank you so much for joining us inside the lab today for this incredibly deep dive into the microscopic, high-stakes world of the PGT biopsy.
It is always a privilege to map out this unseen world.
And our exploration of these microscopic laboratory techniques is far from over. In our next deep dive, episode 17, we are going to explore a closely related micro technique, assisted hatching.
That's a great one.
We'll be diving into the science of the zona pellucida, the tough, protein-based outer shell that encapsulates the early embryo. We will break down exactly why some embryos require mechanical or laser assistance to physically break out of that protective shell, how this highly targeted procedure improves clinical implantation rates, and how it actually serves as a vital required primer step before the biopsy process we discussed today can even be attempted. You won't want to miss it.
Until next time.